All right, good morning, guys. Today we are going to talk about carbohydrates. So, what carbohydrates are is it the fuel and building material for a lot of structures in your body, but also structures in plants as well. So, these include lots of simple sugars like fructose, glucose, sucrose, any of those simple sugars that are either a monosaccharide, so that means that they have one sugar unit, or a disaccharide, which means that they have two sugar units. But that also includes polymers. Anything that's poly means many, mer means part, so many part sugars, which would be things like starch. Every single carbohydrate has a 1 to 2 to 1 ratio of carbon to hydrogen to oxygen, or CH2O, and those can be in any multiples. So if you have something that's C4, that would be H8O4. That would also be a sugar. Lipids, on the other hand, also just have carbon, hydrogen, and oxygen, but the ratio is much, much different. They have many, many more carbon and hydrogens to oxygens and not necessarily a predictable ratio. So anything that we have that is a monosaccharide, those are our single sugar molecules. Disaccharide, di is two, so that is two of them linked together. And then a polysaccharide would be many, many of those stringed together. So if we're looking at monosaccharide monomers, glucose, ribose sugar, all of those things are going to be those monosaccharides. Polysaccharides, these are typically going to be for storage of that chemical energy or for structure. So when we're looking at storage versus structure, if we look at plants, starch, think potatoes, is in the root. That is a very complex carbohydrate molecule. It stores a lot of energy. Where if you look at plant at the structure side of things, cellulose is also made out of carbohydrates, but it is that really tough, rigid material that actually makes up wood. If we look at animals, glycogen is our storage polysaccharide. Okay, so glycogen is typically stored in your muscles and liver for quick bursts of energy when you need it, like if you're running a race or getting chased by a bear or something like that. We as humans don't have structural polysaccharides. However, if you are an insect and listening to this, then you do. Then you have something called chitin, which makes up your exoskeleton. So bugs, arthropods, if they have that exoskeleton, it's actually made of a polysaccharide sugar, and it's called chitin. Words are hard. So the next thing we have is talking about how these things actually link together. So we talked about dehydration synthesis. Okay, so that would also be condensation reaction, because condensation is taking water out of something. So dehydration synthesis, we are taking water out to create, up at the top here, that glycosidic linkage. So that glycosidic linkage is the type of bond that bonds these sugar units together. Just like in proteins, we are going to talk about a peptide bond that bonds them together. In sugars, it's called the glycosidic linkage. Just looking at more examples of this as well, these sugars can link together in different ways. So if you look at the top, we have glucose and glucose forming together to make maltose. Down below, we have glucose and fructose. So if you look at those two molecules, they actually have the same chemical structure. They are both C6H12O6. But when we talked about isomers, we talked about different shapes of things. And if you change the shape of something, then it has a different function. So if you look at the shape of glucose versus fructose, glucose is a hexagon. Words are hard once again. And fructose is a pentagon. So when we're looking at the two shapes of those things, we're seeing a different shape. Therefore, if you have glucose sugar in something versus fructose sugar, the taste of it's still going to be sweet, but the taste is actually slightly different. And it's also different than sucrose. So when you link together a glucose and a fructose, that makes sucrose. Sucrose is table sugar. You guys have all had regular sugar before. If you would taste fructose or glucose versus table sugar or sucrose, it's still going to be sweet, but the sweetness has a slightly different flavor. It's like those artificial sweeteners you taste, how they're still sweet, but they taste a little bit different. Same idea here as well. Okay. When we are drawing out these different things, you saw in the biochem Pogel how we had lots of different ways to draw these molecules. scientists didn't make it easier for you. We have lots of different ways to draw these as well. One of the reasons as well is because glucose and all of these monosaccharides can flip-flop between being that nice circle shape that we see above right here and being a linear form. So they can actually flip-flop and go between, and it's simply the linking and unlinking of those hydrogen groups at the top. So they can link together. See here we have that ketone group at the end, and then when we link together, then it becomes an alcohol group. So we're just linking and unlinking those two different things there. So people that are diabetic have to worry about the buildup of ketones. We're talking about that linear form of glucose. If your body can't store it in your muscles because insulin is that messenger that says, hey, let's suck in all of this sugar and not have it in your blood, you're going to build up your ketones, which is that linear form of glucose. The more you know. Okay. To further confuse every single one of you, glucose also has two forms. It has the alpha form and the beta form. The only difference, if you look at those two pictures, is the linkage of one of the hydroxyl groups. So in the alpha form, if the OH group is attached below the ring, an alpha glucose. If it's attached above, then it's a beta glucose. It doesn't change the function except for when it links to other things, because one glycosidic linkage of these things forms nice little zigzags, and the other one forms like a step. In the polymer form, this can change the function because it changes the shape. Form follows function. We're going to look at different examples of that. So when we're talking about polysaccharides, these molecules are hundreds to thousands of single monosaccharides long. The ones that we use for storage of energy are going to be starch, also called amylose, and glycogen. And structural ones are cellulose, and then chitin should also be on there, but it's really minimal. We don't really talk about it a lot, so we're going to mainly talk about cellulose. So once again, with those glycosidic linkages, up here on the top, starch and glycogen, you can see how that oxygen on the bottom forms like a nice little ziggity pattern, okay? If you look on the bottom at cellulose, we have an oxygen down below and then up above, so it creates like big zigzags. What that allows the plant to do is actually cross-link those fibers. So if you look up at the top at the starch, it forms kind of like little spirally loops, and it actually forms these little packets that then create the starch molecules that your plant can use for energy or that you eat, and then your body turns it into glycogen. So with glycogen, we also have those little swirlies, but it's also a big branching molecule just to be able to like connect everything together. And then down below, if you look at cellulose, now we have parallel lines that are cross-linked together. This is what creates the rigidness that helps to hold like a plant together or hold a tree together or all of those different things. So looking at each of these individually, glycogen is stored in your liver, stored in your muscles, your blood circulates it, all of those different things in order to give you energy. This is formed by alpha glucose units. Okay, the only way it actually differs from starch is just the pattern that it gets put together. So our bodies make glycogen, with the glucose that we eat. Only way it differs from starch is just the pattern, because you're not a potato. I mean, I don't think you're a potato. So starch, this is how plants store that energy. Also alpha glucose can be unbranched or branched. So we have two different forms of that. We have amylose, which is the unbranched version, or amylopectin, which is the branched version. Amylopectin is actually really, really important if you are a jam or jelly maker. So I know during quarantine, quite a few people have gotten into canning. Pectin is actually the carbohydrate in a lot of fruits that help it to gel once you cook it, because it's branched. When it gets heated up, those branches like get all tangled together, and that's what actually makes jelly gel together. So if you don't have enough pectin or that type of starch in your like jelly solution, it's not going to gel and it's going to be like liquidy. Still going to taste great, just going to be really liquidy. So if you go to the grocery store, you can actually buy pectin, which is just this type of starch, and it makes your jelly jelly. Kind of cool stuff. So if we look at this picture over on the side here, these little dark circles and the light circles in there are all little tiny plastids, like little packages, I think of plastids as, with starch granules, starch wrapped all up inside of them. Okay. So once again, oops, got ahead of myself. This is the structure for amylose or starch. Nice little repeating units over and over again. If we look at cellulose with that, these are made with beta glucose. Your body cannot digest beta glucose. This is why we see corn in our poop, friends. This is the reason. We look at those glycosidic linkages. This little oxygen's up, this one's down, this one's up, next one's going to be down. That allows this cross-linking of these hydroxyl groups because we know those hydroxyl groups are polar. The oxygen really loves this hydrogen. It's like, give me that hydrogen, connects together, kicks this hydrogen off, makes cellulose. Okay, so it is a major component of plant cell walls. Most abundant organic compound found on Earth. Reason why? Trees. All the trees. And grass and all the trees. It's a polymer of glucose, but they're in that beta configuration. Cellulose is always going to be straight. This is why we have lumber and wood and stuff like that. So kind of cool little fun facts. So once again, can you tell this is important? We've said this multiple times. Starch is alpha glucose, cellulose is beta glucose. And you're going to explore this more in your homework for today as well. Can you tell this is important? We're going over it again. Cool fun fact, alpha glucose and beta glucose flip-flop. They can flip-flop in solution because of where this ketone group is and where these hydroxyl groups are. They can flip-flop when they're in solution between forms. That's how plants can have both forms of these different things. Lastly, when we're looking at chitin, that is that polysaccharide that is used in arthropods to help build their outsides. So if we would have like a big sheet of chitin, it's going to be really, really leathery, a little bit stretchy, but more just kind of like tough and flexible. When we have chitin that's encrusted with calcium carbonate, then we get really, really hard shells on some of our organisms as well. Because it is really flexible, fungi, think mushrooms, also use chitin in their cell walls. A lot of people a long time ago thought that fungi were plants, but we know they're not plants because I've never bitten into a crunchy mushroom in my entire life. That would be very disturbing. So their cell walls aren't made of cellulose like on plants. They're actually made of chitin, very, very flexible. So the reason why they're so flexible is because this one has a little nitrogen right there. Look at that cute little nitrogen. Oh my gosh, so cute. So a lot of people don't necessarily classify these with other carbohydrates because typically carbohydrates are just CHO. Add in a little, like one little nitrogen, and it just changes the whole structure of this molecule. But we are still going to classify it as a carbohydrate because technically it is a carbohydrate. So that is all for our carbohydrate notes for today. I hope you guys have a great day.